Hi, in this demo, we are going to introduce the web portal developed at J. Craig Venter Institute for RNA-seq data analysis. This is to address the computational challenges in NGS data analysis, which remains a major bottleneck in many projects. So again, here is the URL of the portal. You can also Google JCVI RNA-seq portal to get the access to the portal. This is the homepage of our RNA-seq project. To access the web portal, click this link. So this is the RNA-seq web portal implemented with Galaxy. After login, a user should see a page like this. So on the left panel, there's links to the workflows. The portal offers 3 integrated sets of workflows that can perform end-to-end RNA-seq data analysis. The first set is tuxedo workflows using TopHat, cufflink, cuffmerge, cuffdiff suite of tools. The second set is STAR, mapping and post-analysis workflow, and also we offer Trinity assembly workflow. So all the workflows support pair-ended reads as input and also single-end input formats. So the middle of the page is the summary of the portal and help information. On the right hand, this is user's history and data. So I have run a lot of workflows, therefore has a big list of data and result file. To start using the workflows, user need to complete the registration process, which is very simple. To register, you need to click here. I first need to log out and then hit register. Simply supply the email address and password. Then you'll be registered. So I need to log in back again. Log in. Okay, I'm back in. We provide 2 sets of testing data for users to test the workflows. They're available by clicked shared data, then data libraries. So users can use those data to test our workflows. We provide 2 sets of testing data. One set is from fruit fly, another set is the yeast. Both are RNA-seq dataset. Let's check this out. So those are the FASTQ files. So I click here. That will check all the dataset. So from here, we just choose import to current history. And then if we click go, all those dataset, shared dataset, will be imported in user history. So I already imported also dataset in my history. As you can see, those from here are the shared dataset. So you can use those dataset to run all the workflows here. To analyze your own data, you also need to get them into your history. So from left side, get data, then click upload files from your computer. You can choose local file. Okay. Open. So this will be loaded here. The workflows support FASTQ format data. This is the only data that we support. The files can be compressed, for example, in .gz format. This will be faster to download. Also, for pair-ended FASTQ files, the R1 and R2 need to be in separate files, such as this. One more thing to emphasize is that we need specify the format. So FASTQ and then you push this button start, it will start uploading to the portal. So I'm not uploading it right now. So it may take a while to upload, but then will end up at your history, which can be used to run workflows. So now we are ready to start testing workflow. Again here. So we're going to run the star PE workflow. Click this one. So all the workflows in this portal support multiple samples and the multiple sample groups. So a simple workflow, one workflow run can complete all the major compute for a whole RNA-seq project. The first thing is to choose a reference genome. We support all the major agriculture animals in our database. We also have human and a number of model organisms for testing purpose. So I'm choosing Drosophila fruit flies as reference genome. So for the first sample, I need to give a unique sample ID. I'll choose fly 1 and the label as group 1. So for the R1 reads, R2 reads for sample 1, and I'm going to analyze 4 samples for 2 groups. So click this button to add another sample. So still group 1, fly 2, okay. So I have 2 samples in group 1. Now I'm going to add another sample for the second group. So choose group 2. I'll name it fly 1 control. And another one. So this is also group 2. Okay. So in this workflow, I'll run 4 samples. Each one has 2 FASTQ files, R1 and R2. So they belong to 2 groups. So at the end of the workflow, all the all the compute will be done for all those samples, and will also run differential analysis between the two groups. So I hit execute. That will submit the workflow to the queue. So it may take a while. We are not going to wait, but I'm going to use a finished workflow to demonstrate the result. So this one is one workflow that I submitted earlier. And to access the result, click this icon. It shows up here. I click this link. This will bring up a new page which lists all the results from the workflow. So on the top of the page, there's a brief summary that describes output and the format. So user can download a zipped file that contains all the results from our cloud. Another way to access the result is to browse the whole directory from the to access individual files. I click here. So this page showed all the results from the workflow for every samples, and also has the result of a comparison between the sample groups. For example, let's see sample 1, flight 1. So I have the RSEM result, as you can see here. I have the alignment BAM file here. So for all the 4 samples, I have all those files. This is for sample flight 2, control 1, also flight 2 control. So there are post-analysis of the mapping for comparing the 2 sample groups. So I'm using HR for this comparison. You can see, for example, the differential analysis of genes. You can also see some of the other result like that, and also some analysis from R. So basically, those are the result user can download or browse, analyze individually. Let's go back to Portal. So to learn more about the web portal, we have a wiki page. So this documentation will bring you to the wiki page, which provides most up-to-date information and detailed usage. And to contact us, on this page there's a web form, so you can send a message to us. Okay, this is the demo. Finally, I would like to mention that this project is supported by the United States Department of Agriculture, National Institute of Food and Agriculture. Thank you very much.